Thanks to the organizers for this opportunity. My name is Andres Gutierrez. I am a computational immunologist at EpiVax, and today I will talk about a T-cell epitope content comparison analysis between Porcine Circovirus type 2 vaccines and field strengths. EPIC is the acronym that I will use to refer to the T-cell epitope content comparison. As you may know, Porcine Circovirus type 2 is associated to many diseases, globally called Porcine Circovirus disease. It has been a threat to the swine industry since the early 90s, when it was first reported to be associated with porcine multisystemic wasting syndrome. PCV disease has become one of the most economically important emerging diseases worldwide, affecting pigs primarily between 5 to 18 weeks of age, with mortality rates up to 30%. PCV2 is a non-enveloped single-stranded DNA virus. Its genome encodes two major proteins, replicase encoded by ORF1 and capsid encoded by ORF2. 6 PCV2 genotypes have been described, PCV2A to PCV2F, and there are another 2 genotypes that have been proposed that include potential recombinant forms. A striking feature of PCV2 is its high rate of evolution. Despite being a DNA virus, PCV2 has an evolutionary rate similar to RNA viruses. In addition to the high rate of point mutations, PCV2 viruses recombine. Approximately between 20 to 35% of the PCV2 viruses are recombinants. And since the identification of PCV2A, 2 major changes in prevalence of circulating strains have occurred. PCV2A was displaced as the predominant circulating genotype by PCV2V, and PCV2B was later displaced by PCV2D, which is the current predominant genotype in many geographies. PCV2A, B, and D continue to circulate. Studies in PCV2 phylogeny suggest 2 major groups of PCV2, namely PCV2A and PCV2BD genogroups. As you will expect, vaccination is one of the principal strategies to decrease the impact of PCV2 in the field. Most commercial vaccines are based on PCV2A isolates. Some cross-protection has been demonstrated against PCV2V and PCV2D. PCV2A-based vaccines generate neutralizing antibodies that cross-react and are efficacious in avoiding clinical disease produced by heterologous strains. But there are some concerns about their ability to prevent viral replication or transmission. In addition to humoral responses, PCV2 vaccines also generate cellular immune responses. It has been shown that minor variations in both B-cell epitopes and T-cell epitopes in ORF2 between vaccines and challenge strains result in differences in immune recognition. As PCV2 continues to evolve, the genetic gap between available vaccines and field strains grows. So in this analysis, our goal was to perform a T-cell epitope content comparison analysis to quantify the relatedness between PCV2 vaccines and field strains. Vaccines that contain T-cell epitopes well matched to circulating strains are more likely to induce strong T-cell mediated memory responses upon challenge or field exposure. The goal of this EPIC analysis is first to identify putative T-cell epitopes in vaccines and field strains and then compare them. Based on this comparison, we calculate a score that quantifies how much T-cell epitope content is shared between the vaccines and the strains. That's the EPIC score. Our T-cell epitope content comparison analysis was focused on ORF2, which encodes the capsid protein. We analyzed 4 vaccines, 1 commercial bivalent vaccine, a combination of PCV2A and PCV2V chimeric viruses. This is vaccine AB, and 3 PCV2A-based vaccines. First, the PCV2A component of the bivalent vaccine, vaccine A, and 2 baculovirus-expressed commercial vaccines, vaccine ALT1 and vaccine ALT2. In terms of field strains, we analyzed 614 ORF2 sequences from viruses isolated from America, Asia, and Europe. 108 were PCV2A, 111 PCV2B, and 395 were PCV2D. So for this analysis, each input sequence, vaccines and filostrains, was screened to identify potential T-cell epitopes. The tool we use for T-cell epitope predictions is called PIC matrix. These predictions, the T-cell epitope content, become the input for the T-cell epitope content comparison analysis. The outputs are baseline scores that quantify the T-cell epitope content of each strain, EPIC scores that quantify how much T-cell epitope content is shared between vaccines and circulating strains, and T-cell epitope coverage that assess what percentage of the T-cell epitope content in each strain is covered by the vaccine. To visualize EPIC scores, we use radar plots. Each axis corresponds to one strain. The open circles are their baseline scores, so how much T-cell epitope content each strain contains. The EPIC scores are displayed as the distance along each axis. Each point represents how much T-cell epitope content is shared between a given vaccine and a strain. A perfect vaccine, a vaccine that matches all the T-cell epitope content in the filostrains, will have EPIC scores that are identical to the baseline scores. And we can visualize and compare multiple vaccines. We have the vaccine in gray that has lower EPIC scores than the blue and orange vaccines. The orange vaccine has the highest EPIC scores, which means that shares more T-cell epitope content with these filostrains than the other two vaccines. This radar plot shows EPIC scores for the 4 vaccines we analyzed. It compares 3 monovalent PCV2A vaccines and 1 bivalent vaccine against 614 PCV2 filostrains from genotypes A, B, and D. The bivalent vaccine is shown in orange, vaccine A is blue, VaxAlt is gray, and VaxAlt2 is green. Filostrains are sorted by genotype and by geographical region. Strains from America, Asia, and Europe are grouped, and they are colored in different shades of blue. The results in the radar plot are summarized in this table, where the scores are colored from low in blue to high in orange. The table shows average baseline scores, which are the highest, and the EPIC scores for the strains grouped by genotype and location, as well as an overall summary at the top of the table, considering all the strains by region and globally. And overall, vaccine AB had the highest EPIC scores. For PCV2A filostrains, vaccine AB had a slightly higher EPIC scores than vaccine A or the other PCV2A based vaccines. You can see that in the radar plot, and you can also see that in the table. This result indicates that there are a few putative T-cell epitopes in PCV2A filostrains that are matched by the PCV2B component of vaccine AB, producing these slightly higher EPIC scores for the bivalent vaccine. So in other words, the PCV2B component of the bivalent vaccine adds to the total EPIC score. For PCV2B and PCV2D strains, vaccine AB scores are clearly higher than scores for PCV2A vaccines. These are the results for T-cell epitope content coverage. The percentage of coverage is shown in the y-axis. The vaccines are in the x-axis, and the panels show results for filostrains by geographical region, shown vertically, and genotypes, shown horizontally, including global results by genotype, shown in the first column. The vaccine AB coverage is significantly higher than coverage of the monovalent vaccines, in particular for PCV2B and PCV2D strains. Overall, vaccine AB putative epitopes covered on average 82% of the total epitope content predicted in filostrains. If we consider all the sequences, vaccine AV T-cell epitope coverage was 13% higher than vaccine A, 16% higher than vaccine ALT1, and 19% higher than vaccine ALT2. To summarize, in this analysis, we quantified the T-cell epitope content share between PCV2 vaccines and field strains. On average, vaccine AV had the highest EpiCC scores globally and by geographical region. Vaccine AV also had higher EpiCC scores than the monovalent vaccines for field strains from each genotype, in particular for PCV2D strains that are the most prevalent genotype. And globally, and considering all the analyzed strains, the bivalent vaccine covered on average 82% of the putative T-cell epitope content in field strains. In conclusion, the bivalent PCV2 vaccine had greater T-cell epitope overlap with field strains than monovalent PCV2A vaccines, suggesting that the bivalent vaccine might offer broader T-cell mediated immune response against circulating PCV2 strains. In addition to the application presented today to identify commercial vaccines that might confer the broadest cross-reactive cell-mediated immunity across field strains, EpiC can also be applied to identify vaccine candidates with the highest T-cell epitope coverage for a given set of field strains. T-cell epitope evolution and immunosurveillance are other important applications of this tool, which can be used to identify circulating or novel viruses that are distantly related to the current vaccines for further experimental evaluation to determine potential risk of vaccine failure. These publications are related to the development and validation of PIC matrix and EpiC. The most recent publication shows experimental data that supports EpiC predictions in a PCV2 vaccination study. And in the first EpiC paper, we found that EpiC scores calculated for a commercial vaccine were correlated with vaccine efficacy in swine influenza. If you're interested, please take a look at these publications. Finally, I would like to thank the people working on this project. From Zoetis, Drs. Álvaro Aldaz, José Angulo, Megan Vandrick, and Dennis Voss, and from EpiVax, Dr. Andy Groot, William Martin, and Francis Terry. And thank you all for your attention.